Welcome back to another video tutorial on humoral immunity. Okay, so in the previous video lectures we studied in detail about what is immunity, the types of immunity like passive immunity, active immunity, and also the characteristics of it. So now today in this video lecture we'll study about a main type of immunity which is called as humoral immunity. So the other name for humoral immunity is B cell immunity. So the meaning of humor here is body fluids. Okay, so why do we call so is because in this immunity mainly antibodies will be produced and antibodies will be circulating in the body fluids, and that's the reason we call it as humoral immunity. Okay, so let's get started. So yes, it is a type of acquired immunity. So what's acquired immunity now? So it's the body's ability to recognize and to fight against the specific pathogens. Okay, so as we all know that, so when we get a natural infection or when the vaccine is introduced into our body, so what happens? Pathogens are entering there. When the pathogen enters our body, so our body will recognize it and it will produce a very specialized type of proteins which is called as antibodies. Okay, and these antibodies are very much specific and they retain memory and they specifically target and kill the organisms. So that is called as acquired immunity. And so it is antibody mediated. So that means yes, here antibodies are the major heroes. So antibodies, once the antibodies are produced in a body, we are safe and antigens can no more harm us. Okay. Yes. Now, humoral immunity is mediated by B lymphocytes. B lymphocytes or B cells. Both are the same. Now, as I told, these antibodies are produced from B cells. Okay. So now, where do you find this B cells? How are B cells produced is, see, as you can see in the picture. So, inside the bone, so as we all know, there will be bone marrow. So, this is the bone marrow. Bone marrow will be there. So inside bone marrow you can find a special type of cells which is called as hematopoietic stem cells. Okay, so this hematopoietic stem cells, no, these are very special cells. So they can form all type of blood cells. So these actually can form all type of blood cells. So these are the one which will produce the B cells. Okay, so first it will produce pro B cells. Pro B cells means progenitor B cells. So that means here, so this B cells are in the training stage. They will not be completely trained and they will not know how to produce antibodies. Okay. So this pro B cells will then become pre B cells. So this pre B cells are partially trained. And then this pre B cells will become immature B cells. Immature B cells are kind of developed but they are not completely developed. And then immature B cells will become mature B cells. Once they are mature, so they will know how to produce the antibodies. Okay, and once when the antibodies are produced, these antibodies will enter your blood, will enter your lymph, and they will target and they will kill the antibodies which are present and which are circulating in your body. This is how your immunity works here. Okay, and then now, what are the key components of the immunity? Is first is antigens. Yes, when the antigens or pathogens enter your body will recognize it. So, and then after the first line of defense, so second line of defense will come into play, and then the B cells will start producing antibodies. Okay, so second component is B cells. B cells is important. So B cells are the one which will produce antibodies. So these are B cells, they will produce antibodies which will specifically go target and kill the antigens. Okay, and this antibodies we also call it as immunoglobulins. Then okay, now let us study step by step how the antigen enters, how the antibodies are produced and how do they kill the pathogens. So step 1 is antigen entry. Now unknowingly a new pathogen enters our body. So now this antigen is triggering the immune response. That means our immune system is getting activated. It is getting to know that some pathogen which is harm to us is entered the body. Okay. Okay. Next is the step 2. Step 2 is the recognition and activation of B cells. So how can B cells get activated and how will they recognize the antigen? Okay, so in this recognition and activation, no, T cells also plays a major role. They are the one which will help the B cells to be activated. Okay, so coming from the beginning now, so now antigen is entered a particular body. Okay, so once when the antigen is entered the body, the first line of defense is the one which will come into play. So like skin, mucous membrane, so all these as we know, so these all will come into play. So they try to kill the pathogens, but when the pathogens are not killed, maybe the dosage of the pathogens is more or they're very strong, so they're not killed. So that's when a special type of cells called as APC, that is antigen presenting cells, will come into play. So this antigen presenting cells may be macrophages, they may be dendritic cells. So these are the types of antigen presenting cells. Okay. Now this macrophages or dendritic cells, what they will do? They will engulf the antigen, so that they will eat up the antigen, and a piece of the antigen will be presented to the T cells. Okay. So these are also called as helper T cells. Now these cells, no, so they will recognize the antigen and they will get activated. So once they are activated, they release a special type of chemical signal which is called as cytokines. So once when the cytokines are produced, these are the signal for B cells. Once when the signal is sent, once when the cytokines is present in your body, so the signal will be sent to the B cells. So that's when the B cells will be activated. Okay, but meanwhile, so when all this process is happening, meanwhile what will the B cells do? So B cells have specific receptors on their surface. So as you can see, there are antibodies which are bound on their surface. So these are called as membrane bound antibodies, and these membrane bound antibodies are called as B cell receptors. And these B cell receptors, no, they are of various shapes. Only a particular type of antibody or a particular shape of the antibody only can bind to a particular antigen. So meanwhile this B cells, no, they will recognize the particular antigen and they will lock them with the help of lock and key mechanism. So which shape is suitable for that particular antigen? So that shape of the antibody will attach the antigens, as you can see, through lock and key mechanism. Meanwhile, here the cytokines are also released, chemical signals are also sent, and the B cells will become completely activated. Okay, so this is how the activation happens. And then, okay, now before step 3, so we got to know that yes, B cells are the one, they first recognize the antigens using receptors and they bind to the antigens. Okay, so and then cytokines from T cells will fully activate them. Okay, and one more point I wanted to include is that B cells also will present the antigen to the T cells. Okay, and then the next step is step 3, so which is activation. So now is, as we know that B cells are activated with the help of T cells. So now after this they undergo clonal expansion. So clonal expansion means multiplication. Now as you can see here, so one B cells will form many B cells. Okay, this is called clonal expansion. And then okay, now the next step is step 4, which is differentiation. Okay, so now after clonal expansion, that is when several activated B cells are produced, so they get differentiated into two types of cells, that is plasma cells and memory B cells. Okay, so plasma cells are called as antibody factory because they are the one who produce antibodies. Okay, and so now the antibodies will enter the blood and the lymph, so they will come in contact with the antigen, they fight with them, they kill them, and so finally they will make us get rid of the infection. Okay, and then the second type of cell is a memory B cells. It retains memory, so that if the pathogen tries to enter a body again, so it will immediately act. So now what happened is, first time when the pathogen entered, it was new, so it underwent all these process, and it took a lot of time for the antibodies to be generated in a body. But the same pathogen, when it enters our body for the second time, so it will immediately act, our body will immediately act, and they will immediately produce antibodies. So why? Is because of this memory B cells. They'll retain memory, they will know this pathogen is already entered our body, so we know the mechanism, so it will respond very quickly the next time. So that's the function of memory B cells. Then okay, so now next is the step 5, it is the antibody action. Now the antibodies are released now. So this antibodies will come in contact with the antigens, and so they destroy the antigens. So actually there are 4 different ways in which the antibodies will act on the antigens. Okay, so the first is neutralization. So neutralization is very important. So neutralization is a process in which it blocks the harmful effects of the pathogens. That means now pathogen is producing some toxins, that is poisons, to harm you, or it wants to enter your cell to destroy the cell components. So then what the antibodies will do here, they block those effects. They stop viruses, or they stop the toxins from entering the cell. They block the cell so that they cannot enter, so that the toxins cannot enter. So by means they will block the infection. Okay, and the second type is opsonization. So opsonization is a method in which the antibodies will go and they will mark the pathogen for destruction. So here antibodies are not directly acting. So instead there are some immune cells like macrophages, neutrophils. We studied them in the previous video lecture. So we also know what are their activities. Okay, so now these immune cells will come into play here. So in opsonization, the antibodies are just tagging. That means it is just telling that, so there is a antigen here, it is harming our body, so it is tagging them, it is showing them. So but the immune cells are coming here and they're acting. So mainly here what happens is, macrophages will recognize them, and macrophages will engulf and eat them, as you can see here. So how macrophage is eating them. Okay, so it is engulfing and eating them, that is opsonization. And then agglutination, as the name tells here, it's clumping. Now the antibodies are going and clumping the antigens over here. So in the body, when there is a large clumps like this, no, it will be easier for the immune cells to recognize and to destroy them. And then the fourth is complement activation. So in complement activation, what happens? Antibodies, no, so they will activate complement proteins. There are specialized proteins in a body called complement proteins. They will activate the complement proteins. So now these proteins, no, so they help to break and they help to kill the pathogens. They create the holes in the pathogen and they kill the pathogens. Okay, so these are the 4 ways in which they will act. Okay, so in simple terms, if I want to tell, the first one is it blocks the infection. Okay, so it is blocking the infection in the sense it is tying the, it's like tying the hands of the criminal. So it's not allowing the toxins or the bacteria to enter inside the cell. In the second way, it is opsonization. It's like marking criminal for arrest. It's just marking, it's tagging, and it's allowing the macrophages to engulf, to swallow and kill it up. In the third, it is agglutination. So it's like gathering the criminals in one place. So it is just clumping up with the antigens so that the immune cells can recognize and destroy them. And in the fourth, that is complement activation, it is destroying chemicals directly, like its antibodies are activating some complement proteins, so that the complement proteins, and then the immune cells are recognizing it, they are creating pores and they are killing the antigens directly. Okay, so these are the 4 ways. Okay, so next is the primary versus the secondary response. So what is primary response here? That is when the pathogen enters your body for the first time, it is called as primary response. And when the same pathogen enters your body for the second time, how your body will respond is the secondary response. I think we know the difference now. So now what happens in the primary response? First time when the body, when the antigen is entered your body, so it is a slow response. It really takes time for the antibodies to get generated. Okay, so it may take several days to several weeks for it to get generated. But in the second immune response, it will be very fast, within few hours to few days. So the antibodies will get generated. Okay, so why? Because as we know, in the first time it is a new pathogen, so the body will not know what kind of pathogen is entered the body. It has to analyze what is it doing, how the toxins are produced, what harm is it creating to your body. So it has to analyze properly, and it has to undergo a long step. So as we studied now, it has to undergo a long step, so wherein the B cells has to get activated, and the plasma cells has to produce, and then slowly the antibodies will level, or the dosage will get increased. So but once the primary response is done, as we all know, the memory cells will be created. So this memory cells will have a long-term memory, so that the second time in the secondary immune response, when the same pathogen enters your body, so it will remember, oh, this pathogen had entered our body previously, and so it knows all the steps which is done, how the antibodies are produced, and immediately it will respond, and within few hours the antibodies will be generated mostly, or sometimes within few days the antibodies will be generated, and it will show a very fast response. Okay. Okay, and then coming to the types of antibodies. So as we were speaking about antibodies are not of one type. So there are 5 different types of antibodies. So each antibodies has different shape, and so they have different functions, and they are produced in the different stages of response. Okay, so we'll study all of them in detail, the shape and the structure in detail. So just I'm going to glance what are the functions of these 5 antibodies now. First is IgG. So Ig, because immunoglobulin, as I told, antibodies are also called as immunoglobulins. So it is immunoglobulin G. So this is the most common one, and so this is produced in the later stages, in the secondary response, for most of the infections. Okay, and it gives long-term immunity, and it is found in the blood. So the main role is strong protection and memory. So that is IgG. And then next is IgM. So these are first produced. Yes, so they are they are produced in the primary response, and they are found in blood. So they show quick response and first defense they are. And then next is IgA. So IgA is mucosal. Mucosal means mainly they are present in the mucosal layer, where mucus is present, like saliva, tears, and all the mucous membrane. So their function is to protect the body surfaces. Okay, and then the next is IgE. So IgE is mainly produced in the during allergies. Okay, mainly when there is parasitic infection, allergies. So they'll be mainly attached to mast cells, and they show the allergic response. And then the last one is IgD. Okay, so this is B cell function. Yes, they are produced along with the B cells in the early stages, and so they are found on B cell surface, and their main role is help in the B cell activation. Okay, so these are the 5 types of antibodies. So in short, if I want to tell, is IgG is most common, and so they produce strong protection and memory. IgM is first produced, and they show quick response. IgA is produced in the mucosal surfaces, like saliva, tears, and they protect the body surfaces. IgE is mainly for allergic response, and IgD is for B cell activation. Okay. So overall, if I want to sum up with the humoral immunity, so yes, it is called B cell immunity. So mainly B cells and antibodies play a major role here. So once the antigen enters your body, B cells will be activated with the help of T cells, and they start producing antibodies. And these antibodies will undergo different steps or different ways, and they protect against the extracellular pathogens. Okay, and the main thing is in humoral immunity is they also produce memory B cells, and so this memory B cells will produce a long-term immunity or long-term memory, so that if the pathogen encounters you the second time, you will be safe and your body will act immediately.